So welcome for all of you. I'm going to switch my slide. Yeah, so first one. So what I would like to present you today is the strategies that we use in our lab to analyze the spatial distribution of nuclear signals. I will try to present it with as many examples as possible, a conceptual example as possible that can perhaps relate to your own work. I'm not going to make a step-by-step tutorial of how we do it because there are different documentation which are available. I want to present you the concept and the tools that we developed called DataViz, that is a downstream tool for image analysis. So perhaps let's start with the context. I think most of this attendance is familiar with the fact that the nucleus is not a simple box containing the genome. It is actually a very complex organelle, which looks like a planet on its own on this nice representation. It has different continents, different oceans, different islands, and probably different volcano or different type of active bodies. And our community, in broad sense, is interested to understand what are the organizing principles in these organelles and how this organizing principle helps to fine-tune genome function, let it be gene expression, genome repair, recombination. So in this quest of understanding the organizing principles, we have to acknowledge there is a new field that has emerged in the past 10 years, now recognized as 3D genomics. And this field of research is actually pluridisciplinary and relies on a number of disciplines, let it be modeling, borrowing to mathematical and biophysical concepts, let it be biological or experimental disciplines in order to probe at the molecular level or at the cytological level, perhaps the position of different nuclear domains, their distribution, but also recently methods to probe the folding and topology of different domains in the genome. And in this approach, especially those aiming at determining positioning and spatial cues in the nucleus or folding and topology features of the genome, microscopy plays a big role. And sorry, I'm trying to animate. And microscopy imaging allows to determine, and sorry, this is only one example, for instance, position of certain loci. I'm giving here just one example in Arabidopsis where a locus coding for light-regulated genes has been now labeled by fluorescent in situ hybridization. And it has been shown by Feng et al. In 2014 that this locus is actually moving from the nuclear interior towards the periphery when the tissue is exposed to red light. So that is what is showed with this graph here, and the increase of the bar here shows that there is an increasing amount of nuclei where this light-regulated locus is moving towards the periphery. So there's basically a movement of genomic region towards the periphery, and this is correlated with transcriptional activation of this locus. So this is one example of a study which elucidates a relationship between gene expression and gene position in the nucleus. But this study has been performed with analyzing 2D images of nuclei, and I think there is a lot of room to expand the approach for this particular study, but many other examples towards 3D. And similarly, the field aiming at understanding folding and topology feature of the genome in 3D is also using microscopy imaging approach, in addition to molecular profiling approach to determine contact regions. And here microscopy imaging approach are very often relying on super resolution microscopy to elucidate the structure of a nanodomain in the genome. So this is an example now in Drosophila, where genomic domain which are enriched in activating mark like H3K4 dimethylation in red, or genome domains that are enriched in a repressive mark like H3K27 trimethylation now they have been specifically imaged at super resolution using fish probes, and their topology and their folding is determined by analyzing the packing density of the signal in a certain volume. So this is only to show you that these are two very important approach in 3D genomics, approach relying on microscopy imaging. And there's a lot of room, as I said before, for expanding the use of 3D microscopy imaging. I think we see a lot of 2D imaging, but 3D microscopy imaging is necessary. So we and other people in the INDEPTH consortium, we indeed apply 3D microscopy imaging to try to resolve the distribution of certain nuclear signal. And what we are also developing is pipelines enabling to quantitatively analyze the distribution of this signal. So now I've just shown you a little animation, which is more of a teaser, but I will go through the technicality now with you. So in a few slides. First, what I would like to discuss with you is actually to clarify rather is when we talk about a spatial distribution of a nuclear signal of interest, I make it very general. With signal of interest, I mean by here, it could be a fluorescently tagged protein. It can be a transcription factor tagged with GFP, or a histone modifier tagged with GFP or equivalent protein. This can be a locus which has been tagged by means of FISH, so fluorescent in situ hybridization, where DNA probes are fluorescently labeled. And signal of interest can also be simply coming from an antibody used to detect either a specific protein, a specific nuclear component, or a histone modification, classically. So once we have these labeled nuclear components, what can we actually possibly measure in 3D? And I think this question is very often not clear. So I think many colleagues do 3D microscopy imaging, but then we tend to get puzzled with these complex images. We don't really know how to analyze them. And that's what I would like to present you today, is how we came around this organization, how we came around answering this question, and which kind of pipeline did we develop to obtain some data. So first to the question, what can we possibly measure? I'll try to make it here very simple. We could simply think of a radial organization of the signal of interest. That means we would like to actually measure the distance to the periphery, the distance of our signal to the periphery, and see whether we have a radial distribution. We could possibly think of what I call a body-centric organization. That would mean, for instance, as our signal distributes in a specific way related to a nuclear body. It can be a photobody, it can be the nucleolus, it can be the chromocenter, it can be any other speckle which is labeled. We can also think of determining the type of domains which are created by the signal. So we have different domains of different size and different number, and of course we can also combine it with the previous question, do they have a specific localization? And we can also refine the analysis by considering perhaps that the signal of interest is distributing along clusters, and in that case we would be interested to determine the distance to neighboring signals. So all of these spatial distribution aspects that I mentioned now schematically, we can determine them after image segmentation. And to gain more information about organizing principle, we ideally want to relate them as well with signal intensity, because signal intensity is a proxy for the amount of the protein or the locus that we are probing. We can relate it with the volume of the signals that we are looking at, because it can tell us about the density inside. So this can relate to signal density as a summary. So I hope that's clear up to now. So sorry if my mouse is moving during the presentation. I tend to move the little webinar window away from my own slide. So there is one very important requirement in this approach, is that signals that we want to analyze has to be discrete. That means we need to be able to identify discrete foci or discrete regions, or there should be a method to actually segment the signal into discrete region or foci. That means it could be that your signal looks for you a little bit diffuse, nevertheless, perhaps with some region in the nucleus where it is stronger. And as soon as you have a bit of contrast in the signal, then that means you would be able to segment it and then to make a proxy and determine a fraction of your signal to be considered as a discrete region. So we have developed a workflow for our case, which I think is generally applicable to many questions, many research questions. We start with nuclei extraction, and for our experiments, we embed them in a little acrylamide gel pad on a microscopic slide. This allows them to process in parallel several slides. We can do immunostaining. In our case, the analysis requires super resolution imaging or near super resolution imaging. We use STED. STED imaging stands for stimulated emission depletion microscopy imaging. On this first image, what you have in red is DNA staining with very apparent chromocenters, and in green you have an immunostaining signal, so that's an antibody signal. In this case, it is staining clearly in little dots. So actually I say clearly, but I will show you in another image that it's not so clearly defined by eye. So in our workflow, what we included are steps for image processing, and image processing includes deconvolution of the image. So it is a restoration of the image and to get rid of some artifacts at imaging. And then we have an important step of segmentation where I call it multi-scale segmentation, because we actually segment different objects of different scale in the image itself. And for this segmentation, there are actually many software available in the community, and there are open source softwares. One of them was presented by Dimitar Prodanov last week. But there are other software, and there are also commercially available software. And here I'm not here to make advertisement for the commercial software. I can only present the software we are using. It is called IMARIS, produced by the company BitPlane. So in this webinar, I'm not going to teach how to use the software, because actually the company has developed very well done tutorials. So online you can find all the tutorials, introduction and how-to tutorials to segment in your image. And INDEPTH has already provided actually a training school. So but end effect is that what we have, we define now the nucleus as an object with a boundary, which is now this gray transparent envelope. We defined chromocenters. For those of you who are familiar with it, you know that it consists in a dense heterochromatin region in the nucleus. And then we also defined what we call nanochromocenters. So you may perhaps never have heard about it, but we realized with the STED microscopy imaging that you can actually visualize small heterochromatin foci, which were not visible with confocal microscopy, but we can see them. They are very well resolved in STED imaging, and we decided to call them nanochromocenters. Correspond to smaller heterochromatin regions, and we also segment them because they are inherent part of the nucleus. And then in our image, we are segmenting the antibody signal. In that case, it corresponds to an antibody recognizing RNA polymerase II. And we defined spots, so little spheres, little bits into the nucleus, which are going to capture the antibody signal. I would like here at that stage to interrupt my PowerPoint presentation and move to IMARIS. If I could, yes, I prepared a little movie.
Celia, let me ask, can I come in? Can
yes.
I come in and ask a question just at this stage? So Christophe has asked,
yes.
He said he could not get the exact difference between body-centric and domain-based organization. Can you explain the difference in a bit more detail? So that might be relevant to what I thought.
Yeah, so what I call body-centric organization is if we consider a nuclear body to organize the signal of your protein of interest. So it could be that your protein of interest is gradually decreasing in intensity away from this body, or on the opposite, it is strongly associated with this body. So body-centric means the nuclear body that you're looking at. It can be chromocenters actually, but it can be a photo with it. It could be that around these chromocenters you have certain proteins that accumulate at high density, and the more further away you move from the chromocenters, the less of this protein you have. That would be what I call body-centric. And then the domain organization would be more if your protein is kind of a free-living species and creates sometimes very large clusters that could be, or yes, very large domains, which are rather the interior of the nucleus, and maybe small domains at the periphery. Here you combine both the analysis of the size of the domains, the number of the domain, and the position, but you don't have this radial relationship anymore. But these are kind of categorical organization which I presented as a more of a scheme for thinking about it, but you can actually combine everything in terms of analysis. I just wanted to present some simple case. Does it answer the question?
I think so. Yeah, that's good. It's more detailed. Yeah.
Okay. Here, I hope that you see now my little video. I start to play it. So I wanted to show you still, so to move away from a flat, dormant PowerPoint slide and show you how it looks like in IMARIS. In IMARIS, we have what we call an arena, which allows to browse through your image collection. You can select the image that you want to analyze. In that case, this is a nucleus stained in magenta for the DNA and in green for an antibody with an antibody. You see that it has this kind of punctuate signal. And then in IMARIS, what we were doing is actually to segment. So I'm moving 3D. So it is already prepared in this animation. You see a folder called segmentation, where I created what we call objects, which represent a nuclear contour, an object which represents the nucleolus, and it allows them to define indeed perhaps a distribution of the signal in relation to the nucleolus. It can be inside or outside. This little orange dot that you see now appearing is an object that we plot inside the nucleus. It's called the center of mass. We actually need it to normalize the distances that we will derive from the signal spots later on. So it's a reference. In this image, we have also segmented now the chromocenters. They appear in purple. So these are the big heterochromatin domains. And these tiny little blue patches are nanochromocenters. And lastly, what now appears in green and yellow are spots which are capturing the antibody signal. You see that the spots are a bit elongated. They are not spheres. This is because there is always a little bit of loss of resolution in the z-axis, and you can never have the nice. I mean, with steady imaging, you still cannot have an isotropic resolution. And now, what I'm showing you on this illustration is that on this animation, sorry, is that this antibody signal now defined as spots. Sorry, I might go back quickly to previous step. Now, each of the objects that have been created in IMARIS, they capture parts of the image, and by capturing part of the image, you can read whatever is inside this little object. So that means you can read the intensity inside the object, you can read the intensity sum, you can read the intensity mean, the standard deviation, and you can also have distance which are derived from the position of the individual spot. So on the left side, what you have is a window called statistics, and it is a list of property that are given by each of the spots, and you can actually use them to, in the case of what I'm going to do in this animation, is you can use the statistics to actually color, for instance, the object. So in that case, I've been pseudo coloring the spot with the intensity, I think it was intensity mean. And you see now this shade of coloring in the nucleus that shows the distribution of different foci in the whole nucleus with different intensity. Now, what you can immediately see is that, well, it's colorful, it's nice looking, but it's impossible to interpret by eye. So we definitely need to have a way to export, to plot the data. And plotting the data is possible in IMARIS, but when you have a lot of image replicate, you cannot plot them all in IMARIS. So you need to have your own software, and that's what I'm going to show you. So on this animation, what I'm showing is that I'm going to pause it very quickly. At this stage of the animation, what I'm showing is that you can actually decide which statistics would you like to analyze. You might be a little confused by the term statistics, because as biologists, we often think of statistics as a test, which is here to evaluate the difference between two population. Here in the software, they use statistics to refer to variables which are actually associated with the object. So variable can be distances, it can be an intensity sum, and as you can see from this list, there is a bunch of variables, and some of them don't speak to you, like for instance displacement or cell cycle duration. These are variables very specific perhaps to time-lapse microscopy, but you can choose in this complex list of variables the ones that you're interested in. You can tick them, so they are available in the preference statistics, and you can choose for the spots or for the surfaces, you can choose the one you want. So very often I go for the intensity description, sum, standard deviation, sum of square, mean. I'm also interested in the shortest distance to surface. That means we can measure the distance of the spots to the nuclear surface, The volume is also, of course, interesting. And the same for the surfaces. So what I call surfaces here, these are names associated for objects which are not spots. So that would be the nucleus. That would be the nucleolus, the chromocenter, and nanochromocenter. These are called surfaces. And this animation now, I'm just showing you that I choose specifically some of the statistics of interest. Yeah. Okay, once this is done, I think this animation is going to resume quickly about it. Yes, we go back to the image, and once we are sure that we selected all the statistics that we're interested in, what we can do is to select the folder in which we placed all the objects that we created, and we can export all at once the data associated with the nucleus, the nucleolus, the chromocenter, and the spots, and so on. What it creates is actually a bunch of files. So now there's a folder. When we click on it, as you can see, there's a bunch of CSV files which are generated. So it means that per microscopy image, after you spend time, now it's finished, after you spend time segmenting it, so I need to go out. What you have is a folder with really a lot of data, and that's where the next challenge is starting. So perhaps just also a practical information for you, this step of deconvolution and segmentation with a good image, and if you really know how to do it and what you want to do, you have 2 hours per image, I would say, of work. And when you do it for the first time, then it might be a little bit longer. So it's still some work. Some of you might ask, yes, can you do batch processing? I would say, yes, I tried, but the images are so different that batch processing is not very reliable. So I prefer spending 1 week just segmenting all my images, just follow a protocol, and then it's quite robust. So once you're done with it, what you have is really a lot of data. I'm going to come back to the type of statistics. I would like to shortly mention here that what you should also consider in your data analysis are the different levels. And it's important to think ahead what you would like to compare in the future. If you want to compare genotypes, if you want to compare treatments, of course you want to consider image replicate. So these are different data levels that you need to consider for actually analyzing and organizing your data. So I have to admit that up to a year ago, we were really stuck with this huge amount of data, and we thought, well, we can just write a script in R or script in Python and we plot things. But I'm going to go to, and then maybe I tell you. So at that stage where we thought it's really complex, we realized scripting is not really a nice option, because to explore your data, you always need to write a new line of scripts. You want to compare the intensity sum against perhaps a distance to periphery, or against a distance to the nucleolus, or you want to compare the signal density with the volume. So every time you want to compare to, or you want to plot two variables or one variables, compare genotype, compare treatment, it's, I mean, for those of you who have already scripted in R or Python, unless you're really an expert, it takes a lot of time. That's why I realized we really need to have an interface that allows for interactive plotting of this complex data, because actually what we need to do now is to explore the data. As I said, for many of us, when we are at the microscope, so we have a signal on the screen, but we always have to think about what does it mean and how I can interpret that. And it's the same at this stage. We still don't know what are we exactly looking for, what kind of distribution would be meaningful. So there's still a stage of data exploration which is really important, and therefore data visualization is really important to explore the data, to mature a concept and an idea and a hypothesis. So for this reason, we developed an interface called DataViz. It is still, the refinement is still ongoing, but it is already on GitHub. On my GitHub, I will give you actually the address is written here. If you go to Google and Baroux Lab DataViz, you will have the current version. I just want to put a little note of caution. We are not completely finished. We are still improving it. Yeah, so if you're interested, the best is that you drop me an email, and when it's really ready, I will anyway release a tutorial and put it on GitHub as well. So maybe just one note before I go to DataViz, I wanted to mention another strategic step that we undertook, which was really critical for, I would say, the success of our data analysis. As you can see from these images on the left, which is showing one single plane in the 3D image, it shows you the green signal. In principle, you can guess that they are a discrete region. You can see these little blobs with different intensity. But if you look at them closely, they are always a little bit connected by lower intensity. So it is not very intuitive to think of it as a discrete signal. Nevertheless, we said, well, we can't segment it as a big blob, so we are going to actually probe it throughout the nucleus by using spots. So we are forcing the software to actually find regions in the signals that have a high intensity, and we tell the software, put a little sphere or an ellipse around that, capture it. And we do it in an iterative manner, because the parameters do not allow to capture all the regions with different intensity and different size at once. And depending on the images, in 2 rounds or 3 rounds, we can capture up to 92% of the signal. And the images that you see now in these panels, these are the signals left after each round of segmentation. And you can see, for instance, the first round, you see these little punch holes. These are the signals that was already captured by the first round of spot segmentation. So I took the signal away just to see what is left and what has not been captured. I applied the second round of spot segmentation, and then the third round. And in the end, if you calculate the intensity left, you realize that the spots allowed to capture 92% of the signal, so which I think is reasonable. And for those of you who are interested in the detail, if your signal doesn't have a constant size, then you should use a function in IMARIS called growing spots. So you always start with a minimal size that is available with a resolution of your image. And in our case, that's about 30 times 30 in XY nanometer to 19 in Z, up to 500 nanometers. The 5 is missing. So the spots can be growing from an ellipse of 30 to 90 nanometer up to something with 500 nanometer in Z. So it's adapted, dynamically adapted with a signal. Okay, that was a little detail about segmentation. I'm going back to what can we export from that. So I already warned you before about these statistics, these variables. And now if we think about it, per spot, what we have is, at least in our case, what we're interested in, we have the shortest distance to the nucleus surface. We have the shortest distance to the chromocenter, the distance to the nanochromocenter, the distance to the nucleolus, the distance to the nearest neighbor. So that would be these little schemes. And actually, IMARIS tells you either the distance to the next neighbor or the distance to the average distance to top 3, top 5, or top 9 neighbors. And all this list of statistics, you have it per spot, and you have 1,000 spots or more in your image. And then each spot is also carrying information about the signal intensity. It can be the sum, the mean, standard deviation, and this you have it, of course, per channel. So if you have 2 labeling, like in my case, DNA and antibody called S2P, then I have these variables repeated twice. If you have 3 labelings, you have it 3 times, and so on. And volume is also something of interest. I think here, shape, I put it in gray. It's not so much of interest for our analysis, but it might be for yours. We also extract the coordinate. This could be useful if you want to do more advanced mathematical analysis of the spots distribution. Because DataViz can still concatenate for you all the data, and then you can export a big data frame that you can reuse in your own software. If you don't want to use DataViz, you can still just use it for basically concatenating everything, and then make your own, I don't know, MATLAB, Python, or whatever scripts to analyze it in a more fancy way. So all of this data, this is for each spot, and we're talking about 1,000 spots per, several thousand spots per image. And then for each surfaces, the nucleus, the nucleolus, the chromocenter, the nanochromocenter, or anything else that you defined, photobody, then you have all the shortest distance to something, and all the intensity, sum, mean, standard deviation, and so on. So I think you realize with me that indeed this can be pretty complex, and this is why we have developed DataViz. Now on these PowerPoint slides, and then I'm going live into the interface. On this PowerPoint slide, I'm summarizing what DataViz does. First, it imports the data. It creates a big data frame with all the data that I mentioned before. And remember, all of this data, that's per object, per image, and all your image replicates. So it's really a bunch of data. We have decided that there is a certain architecture. We have levels. We have first a genotype level, then we have treatment level, and then we have image replicate level. Everything is zipped, and DataViz is importing a zipped folder and create a big data frame. It does also a cleaning and reporting, so it's checking the integrity of your data in case, I don't know, you might have forgotten in one image to segment the nucleus center of mass, or maybe you forgot to actually segment the chromocenter. But this does not prevent you from importing this image, but DataViz tells you, just be careful, this image is missing this variable or is missing one channel, so just that you know. So that's a step that we call the cleaning, which is done at the import. I would also like to mention here that in case you're not using IMARIS but another software, if you can create a big data frame which has the same format as DataViz's readings, you can also use DataViz. So we have also implemented the possibility to import a CSV file which would contain the table ready-made. So you're free to concatenate your data the way you want, as long as the format is fitting what DataViz is reading. And then the next step, and it's a bit optional, it depends on user. What we are doing is to process the data a little bit further. We create new variables based on the existing one. So for instance, you might be interested in the ratio between your antibody signal and the DNA signal, or if you have 2 proteins, you would like to know the ratio between the 2 proteins in one spot. So we create a channel ratio. We also calculate signal density. which is the intensity sum normalized. I will come about it in a minute, to the volume. So it tells you a little bit how packed is your signal. What we are also doing is that we are normalizing the intensity per image. So that means in one spot, the intensity sum in one spot is relative to the intensity in the whole nucleus. And we do it for the sum and for the means. Yeah. And then same for the distances. So far the best proxy we found to normalize the distance, not sure it's ideal, but it's still best proxy we could think of. We use the center of mass, also called barycenter. We use the spot at the center of the nucleus and the distance from it to the periphery as a variable to normalize the other distances, at least the distance to the nucleus periphery. At the next step, DataViz allows you to select the data you want to plot, because you might not, if you have 4 channels, for instance, and several treatments and several genotypes and image replicate, you might want to select only a subset of your data first to have maybe a first look at the subset of data. In the future, what we would like to do is also give the possibility to the user to exclude outliers. I forgot to say that at the stage of cleaning and reporting, you also have a reporting of the outliers in your data. And then in the future, we would like to implement a function that allows you to include or exclude the outliers. So you have still the chance to decide on it. So once you selected the data that you would like to plot, there is then an interface which is based on R-Shiny, which allows to interactively plot. So you can choose in pull-down menus which variable you want to plot. So all the variables I presented you in the previous slides are variables that are going to come in a pull-down menu. And then you can choose if you want to plot per image replicate. So you would have one graph per image replicate, or you want to pull all the image belonging to one treatment and you have an average plot, or you can pull per genotype. And you can also then give colors. You can also decide to give different colors per image replicate, per treatment, per genotype. So all the categorical values in your data set can be used to split the plots. It's actually good training for language to split plots, or you can use it to color. And all the continuous variable are the ones that are going to be plotted. And in terms of plot, what we have 1D plots, we have histogram, kernel density, distribution, and classical box plots. And we have also 2D plots, so basically scatter plots. What we also offer the user is a possibility to customize a color palette using the R palettes, and the same as well in the background if you want to have it gray or black if you want some lines. So those of you who are familiar with R, you would find your way around it because you would yes, recognize the different themes. I wrote future ? We would also like to upgrade DataViz to actually provide with some statistical tests, and we still need to define which one and which data, and perhaps PC analysis. I put it in ? because all of this work is depending on funds, and right now looking for funds. So if any of you is interested to contribute and collaborate with that, just let me know. I think there's an interesting future for DataViz. And finally, DataViz is allows you to export the data, either the whole data set or a subset of the data. So basically you started with a bunch of CSV file in several folders. What you would end up with is a unique file, either HTML or CSV file, which is a big data frame. And then you can even reuse it in your own analysis script. If you want to do machine learning based data digging, exploration, then you can, yeah. Okay, so it's a lot of, as a theory at that stage. I will show you still live how it works. I give you just one example, perhaps, of a DataViz can produce. This is one example here with the data from 5 images each for each treatment, A and B. We have normalized intensity mean, and here it's a ratio between antibody to DNA. And then it is plotting this normalized intensity mean along with normalized distance to the surface. So actually the 0 would be the nuclear boundary, and when you go to minus 100% would be the center. And you can see that between two treatments, A and B, then we have a drastically different distribution. So actually the signal is getting has a higher intensity towards the periphery in the treatment B. That's one example, and I think it's really nice to see that, because it's not something we would have guessed by looking at the image. It was far too complex to compute it ourselves with our brain. And another type of perhaps non-intuitive result from data exploration is to look at the relationship between the volume and the intensity in the objects that we are called spots that we capture. And again, we can see that the relationships are differing between the two treatments. So you see that the orange cloud and the blue cloud are different. And again, that's not something that you would actually measure by eye. So maybe if you remember my first slides where I was presenting you the fields of 3D genomics and telling you one of the research objective is to understand the spatial positioning of signal related to expression, for instance. So that would be basically one contribution to it. And we can here look at the spatial positioning of the signal and how it changes by treatment. And on one of my first slides, I also told you about the topology, folding architecture that could contribute. This slide now, packing density, could contribute to this investigation. Even though we are not into fine, super fine scale single molecule imaging, still I think we are approaching, we are contributing to understand the packing and folding of chromatin in that case. Yeah. So before I go to the acknowledgement, I quickly move to DataViz, if you're still with me. I just would like to mention that you basically just need R, where you will be able to copy paste from the GitHub a single line of code. You launch it, and when you launch it, I'm going to increase the window. Yeah. What you have different menu on top, so we are still improving it, including the welcome text and so on. So please consider it as, in a way, it's a work in progress, but I'm happy to share it with you, so it might help you to reflect on your work and see if you're interested. So we have an import tab where we can look for the data file. We can import it. It generates a first data frame. We have a process tab. I thought I had done it in the background, but the process tab is allowing you to to create a normalized data and the channel ratio. Yes, it's going to be difficult for me to process it now. I had to restart it just before the webinar because I've been playing so much with it on my small laptop that it was a bit overwhelming. Okay, and now in the select stage, I can say that I want to use process data. I can remove, actually, in this example data set, I have one image with a little bit of an error. I'm selecting away some unnecessary objects. I can filter the data, so the spinner is still working. So if you have a computer which is a little bit more elaborate than my laptop, it's going to be way more fast than what is showing you now. And we also, I mean, ultimately it would be nice to have it running from a server so that we are not depending anymore on the computer. So yes, so it's lasting a little bit long. Well, if it's not proceeding fast, then I think I will stop here. Anyways, the window 1D plot, 2D plots are allowing the plot that I mentioned before. Maybe I will let it run. Yes, either here that's a density plot. This is a scatter plot, or here's also a scatter plots, but we have the box plot and histogram function. I think I can just complete by saying that you can also work on the, you can actually customize also the limit of the axis, X, Y, you can customize the color. So it's made to be friendly also for the user, so that you can generate plots which are ready to download for your presentation or for your publication, so you can export them. So yes, well, I'm not doing a good advertisement because it's still running, but that's something we are still working out. Perhaps is sometime if you run several instances of R, which I did this morning, you stuff actually your own R session, and you have to restart it from fresh sometime. So my apologies for that, but I think I could deliver the main message about DataViz. I can only say that on a good, powerful computer, it works smooth, very smooth. So perhaps then maybe it's time for question. I still want to acknowledge the people who worked with me on that. So in the past 2 years, Ricardo Rondal, a postdoc in my group, has worked very hard in the experimental pipeline to improve everything until having very nice state images. Recently, a student from Freddy Barnesh lab, Geoffrey Chiv, has also contributed to it. So that's a nice in-depth collaboration also in that case. For state imaging, we have technical assistance from the microscopy facility and the image analysis pipeline. So Rico and myself, we spent some time just to find out the optimal steps for it. And DataViz is actually an interface created by Devin Rouse at the S3IT. It's Computational Science Institute at the University of Zurich. So it's a service that the University of Zurich offers, and based on funds, obviously. So he's been programming the interface based on the concept I designed. And funding comes from different sources, including the Window Foundation, Velux, the little herb suites from Switzerland, Ricola Foundation, and also more classically the National Science Foundation and University of Zurich. So if you have any questions, I'm happy to take them.
Thank you very much, Celia. It's very interesting stuff. It's very impressive as well. So yeah, I would encourage anyone who has questions for Celia about this to please write them into the question box, and we have a couple coming through already. So let me ask, so are you working with IMARIS at all on the DataViz? Are you collaborating with them on that as well?
No, we don't collaborate with the IMARIS people. We use IMARIS for the image processing. So that's the first step after we have the image, and we do it ourselves. I mean, we have a license from the University of Zurich. So we don't collaborate with IMARIS, but I might approach them to tell, well, we developed a tool that could be useful for you guys, and if you want to improve it, you could perhaps support us. Yeah.
Okay, so a question from Tristan, and he asks, he says he doesn't see any data sets available on your GitHub site. Are you planning on uploading one?
Yes, I'm going to apply to upload. I thought we had a data set already uploaded, but yes, it will be. As I mentioned earlier on, if you're interested, best is you drop me an email, and then I will create a little email list of interested people, and then you can get information on the release. I was hoping that for today we were ready with the final version, but there were a few issues coming in between, and well, that's how it is.
Okay. So a question from Axel, and he asks, will you add some of the, I'm not sure entirely what this means, the F and G models from Philip Andre's paper for the analysis of object interaction inside the nucleus?
I would love to, so I know which paper you refer to. That would be in the testing the clustering function. I would love to, but right now it's not on the list. I mean, in the end, it's very pragmatic. So the person who develops the software for me, it's a contract-based service, and I need to have funds basically. So we are nearly finished with the funds I had with them. He's going to finish some current interface. Any future implementation requires more funds. As soon as I have, yes. Yeah, but that would be interesting. Yeah.
Yeah, absolutely. So a question from Christoph. So when you're using the SMR and the immunofluorescence with antibodies, what's the best way to prepare the samples? For example, embedding in acrylamide, or do you have difficulties getting the antibodies to enter the sample?
We don't have so much difficulty to enter the sample in acrylamide. Actually, for us, it works quite well. And we chose acrylamide embedding so that we can retain the 3D structure of the nucleus. And so far, all the preparation mode that we tried were not satisfactory for 3D structure. You just end up in a flat dome. Yeah, so that's the reason for acrylamide. And I think what is more critical for super resolution imaging is a type of antibody, and they have to be adapted to the imaging platform. The fluorophores need to be really resistant to the laser, strong, harsh laser, which are imposed in either STED or STORM. Yeah.
Okay, a question from Ulkar, and he asks, so he's got some mutant lines which have a dispersed chromocenter. He's wondering if it's possible to, via the spot segmentation, to visualize the structure, but without super resolution microscopy.
Okay, if it's about chromocenters, I think there are other software available which do the job very well, and one of them is nucleus-J version 2 that Christophe Tatou presented now a few weeks back, and you don't need super resolution, you need indeed just confocal microscopy, and there are very simple segmentation, I think, interface to do this. So I would refer rather to nucleus-J from Christophe Tatou for what you want to do, if I understood the question properly.
Yeah, no, that's lovely cross-posting there. So if you look on the INDEPTH Academy page, there's a webinar from a few weeks ago from Christophe, where he explained their new version of nucleus-J. Yes,
yeah.
Thanks.
And what Dimitar Prodanov presented last week, it's also an open source algorithm that allows to segment, I think, the chromocenter. I haven't tested it myself, but that would also do the job. Yeah, so there are a few possibilities for that. You can still do it with IMARIS. I don't say you don't have to do it. I just wanted to say there are other possibilities.
Okay, so if anyone has any other questions, please can you write them down? We have a question. Oh, here we go. So, okay. Yes. So Ulka replies, if you could write the software. Unfortunately, he missed the course on that. So yeah, that's it's challenging to use it, but there's plenty of opportunities to obtain IMARIS training through other sources, right?
Yeah, exactly. So BitPlane website have a really excellent learning resource. Just check it out, from basic introduction to introduction per tool per topic. It takes, of course, a little bit of time to get into it, but if you have a real research project that requires image analysis, and it is worth it. And after a week, you're getting good at it. Yeah.
Absolutely. And I will send you a link for the INDEPTH Academy now, in case you missed that as well. So, okay, so I think, Celia, we don't have any other questions coming through, so maybe we will leave it there, if that's okay.
That sounds good.
As Celia said, she's very well. She's happy to receive questions through email. So if you have any questions for her, then please send them, and she'll reply to you as soon as she can, obviously.
Yeah. So questions and interest. If you have interest in DataViz, I will keep a little mailing list so that you get informed of the release stage and which improvements has been made.